Hello everyone, I am Ceren from Koç University, and today I will be presenting my project, which is titled as Network Analysis of Cancer-Associated Phosphoproteomics Data to Reconstruct Kinase Network Topologies. So here's my outline. First, I will briefly talk about network motifs and kinase signaling networks' importance for the project, and then I will explain the methodology that I use to construct my reference network to find significant motifs, and the specificity tests that I use to filter out my patient networks. Then I will give my results for cell line patient correlations, the clustering patients, and the network proximity method that I use to find patient representative drug and drug targets. So network motifs are small patterns of connections that are found higher numbers than in randomized networks. So there are 500 kinases and 200 phosphatases encoded in the human genome, which enable rapid regulation. And the phosphorylation sites play an essential role in these regulations. And kinases mediate this phosphorylation event, and they are found most growth and survival signaling in multicellular organisms. But kinases are often found dysregulated in cancer, and this leads to uncontrolled growth and progression of the disease. So kinase signaling networks have an importance because kinases have the largest fraction of FDA-approved drugs for oncology, and understanding these specific kinase signaling pathways can help us find kinase biomarkers that are specific to patient and patient diseases, and this can lead us to find potential therapeutic targets for targeted therapies. Also, we can find mechanisms of drug resistance and combination therapies to overcome this resistance. So this project is mainly about identifying specific network motifs within ovarian tumor-specific kinase signaling networks, and to find clinically significant target genes or pathways by aligning these patient networks with ovarian cancer cell lines. This can offer us valuable insights for developing personalized treatment strategies by using these kinase inhibitors in a clinically relevant context. So here's the methodology listed that I use. Firstly, I have constructed a reference interactome and found some significant network motifs by comparing my interactome to a random network. And then using these significant motifs, I have constructed my patient networks with using expression thresholds. And then I have filtered out these patient networks with some calculation of a specificity scores. After I obtained my patient networks, I have matched them with selected cell lines and clustered them according to their drug effects on survival. And then I have found drugs that represents my patient groups. So for reference construction, I have used phosphoproteomics data of an ovarian cancer and kinase and phosphatase databases. I have merged these two databases together as sources being kinases and phosphatases and targets being phosphoproteomics data's proteins. After I constructed this big interactome, I have searched for the network motifs that have significant meaning in biological systems. I have compared my reference network with a randomized network with the same node size and the same edge probability with my normal network, and then I found some significant motifs. Afterwards, I have constructed my patient networks according to these motifs, and then I did some specificity tests as node swap and random seed selection to filter out the unnecessary edges and nodes from my patient networks. After I got the final patient networks, I have correlated them with cell lines that are clustered similarly with my patients. I have selected cell lines and their drugs that show sensitive and resistant profiles. Then I have found their drug targets, checked their pathway distribution, and also checked their effect on my patients by doing survival analysis. I have found some of the genes that have a survival effect on my patients. I have checked their expression levels and found a distribution that can be clustered. When I check the distribution in survival, I've seen a significant difference, and I have clustered my patients. And then I use network proximity method to find the drugs that are close to my closest to my patients. And I have taken account of 3,000 drugs and all of my patient networks. And then after I clustered my patients, I have checked their mutation profiles and selected the driver genes on these mutation profiles. And I have select them as a drug targets, and by using network proximity method, I have found drugs that represents my patient clusters and combine them into together. So I have found drugs that represents my patient groups. So as to conclude up, this study highlights the importance of identifying specific kinases and motifs driving tumor growth and potential therapeutic targets for personalized medicine. So this proposed methodology enables to generate patient representative networks, and the correlation of these patient networks with cell lines assist us in the identification of successful treatment options for patients with specific kinase biomarkers. So thank you for listening to me.